Hi, everyone. So I see people joining. So I'll start slowly. So I'm Etai Jacob from AstraZeneca, and I'm going to talk with you today about one of our new methods for discovering predictive biomarkers. So that's using contrastive learning. Right. Okay. So I think now most of the people joined. Tell you a little bit about AstraZeneca. Oncology R&D has a very clear strategy to end cancer as a cause of death. And this strategy includes 3 pillars: attacking cancer from every angle. And in AstraZeneca, we have many therapeutic modalities to do that. And there's highly invested across the portfolio. And you can see those in the left from ADCs to immuno-oncology. Another pillar in our strategy is really dive into the science and have a deep understanding of the tumor microenvironment, and the patient health. And the third, which I'll dive in with one of our methodology today, is really a disruptive data science and AI machine learning development to really interrogate all this data. And when we're talking about data, there where the oncology data science group of hundreds of people comes in, and we work on all modalities and levels of data, from molecular to population level, and for that we need a very diverse set of scientists that will work from bioinformaticians, computational biologists, AI researchers, and so on and so forth. So. And there's several openings. If you're interested, you could always connect with us later. So this is our organization. And in ISMB this year, we have 3 presentations. The first one I'm giving now about AI-based predictive biomarker. And we're going to have on Monday afternoon, we're going to have Martin talking about computational modeling of immune therapy resistance, and Kader talking about imaging models for survival risk to immunotherapy. So the whole 3 talks are about science, so you are welcome to join and hear about our research. So now I'll give you some motivation why we're interested in biomarkers. What you see here, this is the drug R&D development process. Many steps. Each step is very hard, and what you see here is a view from target validation to approval to launch. And you have some interesting information here. Time it takes to develop each step, a cost per new molecular entity, and a probability of success. And importantly, if you look in the middle from phase 1, from the starting of the clinical study, for each 10 molecules we bring in, only 1 would be successful. And not only that, also the time it takes and the money investment is huge. It's actually more than 50% of the whole drug R&D process. So this is our motivation, where we really want to bring biomarkers. And why biomarkers? And what you see here is a summary of a decade of data from clinical trials. And this is a dynamite plot of the probability of success for different transition from one phase to another. So for example, you could see that moving from phase 1 to 2, the probability of success is around 50%. And this is also divided by those studies that have a pre-selected biomarker than those that are not. And interestingly, what you could see that if you have a pre-selected biomarker for your study, your chances of success are 2-fold higher. So this is where we want to really bring the biomarkers. So this is the motivation, and that sets us up really to put a lot of resources into really building this translational effort. And biomarkers can be any measurement from an individual. It could be at a molecular level, cellular level, patient level, and so on and so forth. And you could see here on the bottom right the different measurements that we can take. And these are the bread and butter of the analysis that we are doing across the value chain. So In the last few years, in oncology data science, we have developed several tools to tackle the problem of biomarker discovery. Here I'm just indicating a few of those that we have developed that are available in the public domain. We have several others that are not, but in the public domain we have this, the biomarker navigator, PressNet, immune-mediated adverse events signature that we execute across more than 6,000 patients, the clinical transformer that is based on sort of a foundation model based on transformers, multimodal data integration. What I'm going to talk today is the predictive biomarker modeling framework. What is predictive biomarker? So a predictive biomarkers define a subpopulation with strong tendency to respond to a specific treatment. So if we start on the left in a clinical trial, all the patients you see is in the same color, and we define two populations, one B plus and one B minus. The B plus would be those patients that have that characteristics, for example, EGFR mutated patients, right? That could be defined as a biomarker. And if those, and the patients that have a wild type, those would be defined as the B minus, those that do not have that characteristics. So if we separate the population based on these two, and we have this green and black colors, and then we look at the two treatments, and treatment could be immuno-oncology, one treatment, another treatment could be, for instance, chemo or any other standard of care. It could be also placebo in a clinical trial, right? So then what happens is that on the top right, you see that those biomarker positive patients would respond much better than the biomarker negative that you see in the dashed line for treatment one. And this is a standard survival curve, survival plot with on the y-axis you have the survival probability and the x-axis is the time. So what you can see here, treatment one, patients with the treatment that receive the treatment and are biomarker positive are surviving much longer than those that are biomarker negative. On the other end, on treatment two, you see that regardless if they're biomarker negative or positive, still there's no added value there. So that's a predictive biomarker, and now let's talk about prognostic biomarker because the difference is essential to understand. So what you see here on the top is again just the whole population without stratification, and we see that those that receive the treatment, the curves is above the dotted line, the placebo, meaning that the treatment benefits those patients, and on the bottom left you see the prognostic biomarker. So if you look at the black curve of the versus the green one, then you see the biomarker positive versus the biomarker negative, and you see that there is value for those patients with biomarker positive. Now, if you look at the red placebo, biomarker positive versus the blue biomarker negative placebo, we see that also those patients benefit that are B plus, that are biomarker positive, benefit from the treatment. So a prognostic biomarker is defined as a clinical biological characteristic that provides information on the most probable patient health outcome, irrespective of the treatment. So and so this is a very important distinction, right? Where the predictive biomarker would be enable information. This will enable us to understand those patients that benefit from the treatment. So this distinction guides us to develop this methodology, and really looking for those predictive biomarker is a very challenging, and usually the data you have is only single arm, only one treatment, and then the biomarker you find usually would be could not be translated to other indications or other treatments. Right, so for example, age is a pretty strong prognostic biomarker, but age also, regardless of the treatment you get, will say something about your survival probability. So single arm analysis tend to identify biomarker with prognostic value, and single arm survival analysis often overestimates the predictive potential. When we have data of two arms, then we can do different things to identify a predictive biomarker. A standard approach would be a grid search at various quantiles, examinations of the Kaplan-Meierkers, and then looking at the p-value and trying to make a decision. This approach is challenging, and when you think about high-dimensional spaces where you have multiple variables and you want to find a subset, then it becomes really challenging. So, and then obviously the most important thing is, and the greatest problem is this overfitting. So overfitting possesses a significant challenge in biomarker discovery, mainly due to heterogeneity in patient populations and large number of features. But these are really things that we all encounter and have to deal with on a daily basis. And in the method we have developed, and this is just a recap about what is contrastive learning in very high level. Contrastive learning involves training a model to differentiate between similar and dissimilar pairs of data points by maximizing the similarity within the same class and minimizing it between different classes. On the left and right, you see moving from the initial embeddings to the trained one. You could see that the a negative pair of a dog and a cat, and then the positive pair would be the anchor dog and the other dog, augmented dog. So, and as you could see, the vector length, the distance between the negative pair is longer, is higher than those of the positive, and this is the idea behind the contrastive learning, and we take this idea to our method, and this is the high-level core idea behind the algorithm. We could frame it as a clustering-guided contrastive learning. So first, we label the patients to B+ and B- biomarker based on the input, and then the second step, we create pairs of patient groups. So the positive group pair that I showed you earlier, the positive would be, for instance, the dog and the dog, would be B+ and B- patients from the control arm, and the negative group pair would be B+ and B- patients from the treatment arm. And then at the third step, we train a model using contrastive loss, where we maximize the distance between the negative group pairs, so the treatment arm, as you can see here on the bottom left, and we minimize the distance between the positive group pairs, those that are in the control arm. So this is the idea behind the algorithm, and you see the loss function gamma 2 divided by gamma 1 plus some prevalence component that we add there. And this, and now I'm going to show you some results. But before that, just to see how it looks like in the training. So this is as a function. What you see is a function of epochs, and you see is that the treatment, in this case, immuno-oncology, we see that the difference between the curves increases, and when the control, it keeps staying the same. So we try to build a model that will be able to find a distinction between two patient populations on the treatment in the context of not making any difference in the control. So this is the idea. Of course, what you see here is the training, and this is a summary of this part. So from patient population labeling and the training model, and now I want to show you an example how it works, and I'm just gonna tell you in a bit now on the data we're using. So here we're using publicly available data that you could all download of Poplar and Oak that are phase 2 and phase 3 respectively clinical studies of immune checkpoint inhibitors, anti-PD-L1, atezo, and then the control arm, the standard of care would be chemo. The Poplar, so Poplar is this phase 2 is around 300 patients, and the Oak is around 1,200, around for these two arms. And what you could see here on the right is the final publication of that study, where you have a successful trial with a hazard ratio of 0.75 approximately. This is, by the way, not AstraZeneca trial. It's just publicly available, and it's organized in a way that can enable you to do your research in a productive way. And so what features have been using to feed this model that I showed you earlier? Around 30 features, 10 demographic clinical, and around 20 from the top most prevalent genes, mutated genes, so around 30 per patient. So using this table, we feed the model, and we came up with these results. And what you see here on the left, this is the training, and the comparison you should look at is the red versus the orange, which is a very strong, of course, it's overfitted because this is a training. So we have a very strong separation in the atezo, in the treatment, and then the standard of care, the control, we see that there is no separation. So the model was able to successfully predict this predictive biomarker. And then if we move to the right, we see the test. So OAK is the phase 3. It contains 1,200 patients versus the around 250 we have in the other one. And we can see that there is a consistency within the biomarker where we have a better way to predict we're doing better in finding those populations that would benefit from the treatment. But that's not the end, right? Because in clinical settings, it's important to understand what's behind the signatures, what can help us select the patients and understand that. And this is why we have the model distillation part that translates the neural network to a clinical decision tree. And the way we do it, so the model I showed you earlier comes up with one neural network. So here we're training 100 different neural networks with the same system with some randomization. We take this ensemble and we do some refining and choosing those that we believe more. And then on those, we label the data and we take those with high confidence that you can see in the middle plot. Now, once we have a labeling and everything is done only on the training set, now we do pseudo labeling and they use that to train on a single decision tree that you could see here in the middle. And this decision tree is then applied to the OAK, to the test set. And what happens when we do that, you could see that our results are even better. On the top, you see the model I showed you the results I showed you just earlier, and then applying this one single decision tree, we see that now we even have a better ability to pin out patients that would benefit more from the treatment of atezo versus docetaxel. In the bottom, you see the decision tree that actually we found, and what you see here that, for instance, maximal somatic allele frequency, this is the root of the tree, is the main important features, but all of them are required in order to come up with this predictive signature. So this is the idea. And we have executed that across nine clinical studies. One of them I had to hide because it's not published yet, but you could see 8 of those, and in all of them you could see the same tendency of, and this is only the test set of our model to be able to predict, to come up with the predictive biomarker. This is just another way to look at it. I'll skip that to keep some time for questions. This is a summary of the modeling, so you could think about this decision tree that can include different modalities together, from demographic till others. Yeah, so, and this is the research group. The leading author of this work is Gustavo Arango, that you could see here in the thick, under the thick arrow. The second author is Damian Bickel, and then other significant authors, Jerry Sun and Caitlin Smith. And this is the group. Our group is located across the globe, from San Francisco, New York, Boston area, UK, Barcelona, and now we're also recruiting here in Mississauga near Toronto. So thank you, and we have, I think, 2 minutes for questions. Please.
Hi, thanks for the talk. I'm wondering about your feature selection criteria. Did you ever test against the, in the case where there could be no correlation between your marker and the any features that you have for the samples? Do you correct for those sort of bias?
It's a good question. So essentially, the idea is that all features that are, so for instance, in OAK, we have 30 features. All of them are input for the model, and the model do the selection its own way. And then you could look at the tree and see what happens and which ones would be more important, which ones would not. I definitely would state that when you have a correlated associated subset of features, that could have some confounding effect. We are doing simulation to investigate that further. There's other confounding effect, for instance, prognostic features that are also predictive to some extent than others. I think one of the advantages is because this non-linear modeling system, you're able really to pin out relationships that are more complex and be able also to include that kind of dependencies, but this is definitely something that we have to take into account. I think that this method actually has an advantage, even when you have some correlation in the structure of the data.
I have a few more. About your patient-centered stratification, the setting up the threshold for the marker to separate the sample population will be another non-trivial question. Do you think any like more granular modeling will be possible, where you can, for example, for the transcriptomic markers, you can use the not the binarized values, but like do you think we can use the raw values to do such modeling?
Right, raw values. Well, it depends on how much data you have. I would. I mean, the next thing, and that's something we are doing, is actually we're using a sort of embedding space, and then we use that to feed the model I just shown. The thing is that you need to go backward and understand what happens. So then you need to use different methodologies, but that would be the way to go. Using raw data a priori, for instance, transcriptomic would just waste some of the entries on really understanding the patterns within the data. So I think that's why foundation models and other aspects can bring more value. But it's a legitimate question. At some point, we'll have infinite number of samples, and then maybe we could do that. But we are not there yet.
Thank you.
Thanks for the questions.